to welcome Kate Snipes, Minister Counselor for Agricultural Affairs at the US Embassy in Paris, for introducing the well-known Alison van Eenennaam, the genetic scientist at UC Davis. So after the seminar, we had a debate with the audience and a press conference. So I would like to give the floor to Nadine Vivier. She's the Vice President at the Academy of Agriculture, and I would like to wish you a fruitful afternoon.
Well, it's really a great pleasure and an honor for us to welcome here in our Academy Mrs. Kate Snipes and Dr. Alison van Eenennaam. I would like first to thank Kate Snipes to come again. A little more than 1 year ago, you recalled the relations of Jefferson and with Dupont de Nemours and with our Academy. And now we are happy to really maintain cultural exchanges with the US Embassy. So thank you for coming again. And also, I thank Dr. Alison van Eenennaam, who is going to give a presentation of genome editing, cattle genome editing. This is a subject of the working group of the Academy, so a very important subject for us. And we know how important and how passionate advocate you are for this science diffusion, since we have seen the film Food Evolution. So now we are very honored that you gave us the presentation. And last, I am going to thank Agnès Rickrock. C'est grâce à elle que nous pouvons organiser ces séances, resserrer nos liens avec les États-Unis et avoir des personnalités importantes. Donc là, j'ai préféré. Donc, merci. Mrs. Kate Snipes, we are going to listen to you.
En français, je vais essayer. Donc, on verra. Mesdames et messieurs, je remercie l'Académie de l'agriculture de France pour m'avoir invité à introduire la présentation et le débat de cet après-midi. Mais je félicite surtout l'Académie pour avoir invité professeur Van Eenennaam de l'Université de Davis en Californie à venir s'exprimer ici. Dr. Alison est une grande chercheuse, une scientifique reconnue. Qu'elle mène des recherches dans le cadre de son rôle de spécialiste de la vulgarisation agricole au département de zootechnie de l'Université de Californie à Davis, où elle dirige le laboratoire de génomique et de biotechnologie animale, où qu'elle parcourt le monde pour parler des implications de son travail. Professeur Alison n'hésite pas à s'engager à creuser le fer pour que la science et le fait scientifique s'imposent. Ce n'est pas une position facile, étant donné que le sujet des productions animales, comme celui des biotechnologies, déjà controversés individuellement, deviennent complètement explosifs lorsqu'ils sont combinés. Cependant, professeur Van Eenennaam s'est toujours fondé sur des données scientifiques solides, malgré l'hostilité directe sur des sujets allant du clonage à la sélection génomique et passant par les animaux modifiés génétiquement. Nous devons prendre la parole a-t-elle déclaré. Je ne laisserai pas les alarmistes prendre en otage le débat. Des groupes anti-progrès et anti-science ont tenté à plusieurs reprises de faire taire sa voix puissante en menant entre sa réputation scientifique, notamment par le biais de sites internet alternatifs critiquant son travail. Cependant, Dr Alison, grâce à son dynamisme, réussit à entendre le point de vue opposé et sans partager son enthousiasme contagieux pour ces sujets de prédilection et sa compréhension habile des questions pièges. Et puis, il y a son curriculum scientifique impeccable, avec plus de 70 publications scientifiques qui présentent un intérêt pour son domaine, notamment Nature Biology, Genome, Transgenic Research, Journal of Animal Science, Journal of Dairy Science and Metabolic Engineering. Elle a également écrit plus de 130 résumés et comptes rendus d'études et de recherches. Passionnée de science, Dr Alison a reçu le prix Borlaug de la communication du Conseil pour les sciences et la technologie agricole en 2014. En 2017, elle a été élue membre de l'Association américaine pour le progrès de la science, AAAS. Elle siège également au Conseil consultatif de la Cornell Alliance for Science. Plus récemment, on notera sa collaboration à une utilisation pionnière de l'édition génomique visant à rendre silencieuse le gène qui produit des cornes non souhaitées chez les vaches laitières Holstein. Ces études ont été largement commentées dans le milieu scientifique et se intéressent par le bien-être animal. En plus de faire tout cela, elle a également un calendrier de conférences incessant. Ayant déjà présenté plus de 550 présentations à un public dans plus de 2 douzaines de pays. Professeur Alison a également beaucoup utilisé des vidéos et des parodies pour faire connaître au plus grand nombre les avancées de sciences agricoles. Elle a même été une des vedettes à un documentaire Food Evolution où elle défend les biotechnologies de manière si élégante qu'elle a incité certains à changer de vie sur la question des gènes. Bien qu'elle soit engagée dans ce débat depuis plus de 2 décennies, Professeur Van Eenennaam reste toujours déconcerté par l'antipathie, pour ne pas dire plus, que suscite son domaine de compétence chez certains opposants et critiques. En tant que mère, Dr Alison avoue être profondément troublée par le fait que plus de 25 000 personnes, des enfants pour la plupart, meurent de faim chaque jour dans le monde. J'aimerais faire partie de la solution pour que cela ne se produise plus à l'avenir, disent-elles souvent. Je remercie donc chaleureusement professeur Van Eenennaam pour venir dans cette enceinte pour nous parler de ses recherches et de ce qu'il se fait dans le domaine de la génomique animale aux États-Unis. Je ne doute pas qu'elle trouvera ici un auditoire passionné et intéressé à même de faire partager son savoir et ses expériences. Merci aussi pour toutes les invitations, les deux. C'est mon plaisir. Malheureusement, j'ai en même temps une grande délégation de producteurs américains de soja qui sont à Paris aujourd'hui, mais je vais rester avec vous aussi longtemps que possible. Je vous remercie et laisse parole à professeur Van Eenennaam. Merci.
Merci et bravo pour avoir fait ce discours dans un salon français.
Thank you for that introduction, although I don't speak French, but it sounded right. And thank you for the invitation and for your attendance here this afternoon. I'm going to talk about gene editing in food animals and focus a lot on cows because I like cows. I think cows are amazing animals, taking inedible cellulose and making nutritious food. And there are cows throughout the world in different geographics that are suited to those environments. And we see here a range of them, including a time when I was a bull rider, which you may not have known I was able to do. He was a very gentle bull. But we have done a lot of genomic analysis on bulls. In fact, some of the people in the room here have been involved in this work. And a coalition called the 1,000 Bulls Genome Project has now resequenced over 2,700 different bulls from all the breeds on Earth, healthy males with offspring, and we find there's in excess of 86.5 million genetic variations between those bulls, SNPs, indels, that give us the amazing genetic diversity that we have in this breed and also the adaptations that we see in these different animals. One's adapted to tropical conditions and one's adapted to temperate conditions. And breeders use this variation to produce animals that have characteristics they want. And of course I lead off here with a French poodle, how appropriate, but also we can see many variations from the ancestor, the wolf. Breeders have selected for animals based on their appearance, according to what the breeder is looking for. And some people think these are very nice dogs. And perhaps there's some truth to this slide when you go home and look at your dog tonight and see if there's any truth to this slide. But when we start applying intentional selection to our agricultural organisms, that is when it becomes really important to our overall food production and agricultural sustainability. And we see here maize,
over time, starting in 1865 through the current time. And what we see is yield is consistent, flat, until a new innovation comes along. And we see an inflection point, a rate change in the improvement of yield per hectare. And the more yield we can get per hectare, obviously that reduces the overall environmental footprint of a kilogram of maize. We can see a similar thing when we look at our animal production. And here we see dairy cow yield in milk kilograms per cow per year, going along flat, like the maize. But then, in 1944, we have the introduction of artificial insemination. It enables the best males to be used more heavily, and so we see the increase in yield. And basically, the way that breeders think about this is we are always trying to increase the genetic change per year that we can accomplish when we select the best males to mate with the best females. And there are 4 components to the breeder's equation. There's the accuracy. How certain are we that he is the best bull? The intensity of selection. How much do we able to use the very best genetics and basically distribute the best genetics over a wide number of matings? The amount of genetic variation we have available in our population, which gets back to that diversity. If we have a population of clones, we have no genetic variation, so we cannot make genetic progress. So that's an important thing to maintain. And all of that is inversely proportional to generation interval. That is the age at which you become a parent. And in cattle, biologically, that's about 2 years of age. But typically, we have bred bulls later than that, after they have proven themselves as sires through progeny testing programs. And recently, we've had the opportunity to actually use the genomic understanding of our bulls to actually do a thing called genomic selection, where we genotype bulls, and based on known relationships between genotypes and phenotypes or measurements, we're able to get genetic predictions on the genetic merit of young bulls, and we don't have to do progeny testing anymore. And what that has enabled is basically a doubling in the rate of genetic gain towards our selection objective, which is, in this case, net merit. And this is another inflection point that has been enabled by the sequencing of all of these bovine genomes and the application of our understanding of quantitative genetics married with our phenotypes or databases that has enabled that to occur throughout the globe, basically. And it's amazing to me, it's only 2019. This technology was introduced in 2009. So in 1 decade, we've doubled the rate of genetic gain just using better genomic information. And so what's the impact of all of this genetic improvement on the overall environmental footprint of our food production? And this is where I want to kind of bring out the importance of genetics to sustainability. If we look at the dairy cattle numbers in the United States back in 1944, when artificial insemination was first introduced, there were 26 million cows in the US. Today, there's 9 million cows. But they're producing about 1.6 times more milk than those 26 million were back in 1944. And that has a very big impact on the environmental footprint of a glass of milk. And that's actually being quantified by this paper, where you see the milk production per cow is over 400% increase, a 4-fold increase. But what that has enabled is less animals eating less food, drinking less water, on less land, producing less manure, such that ultimately the environmental footprint or carbon footprint of a glass of milk today is 1/3 what it was in 1944. And that's very often not thought about when we talk about genetics and breeding. It's really an important overall concept in sustainability. And beef cattle have a similar story to tell. They haven't had quite the straight path that the dairy cattle have, because there was a period in the 50s when people were selecting for very little cows, and you can see them there in the Grand Champions. And then the breeders decided that they wanted bigger cows, and so in the 1980s you see these very tall beef cows, which are probably not optimal because they're going to be quite hungry, and they probably are not an optimal animal. And so it's a little bit like Goldilocks. It was a little too small and a little too tall, and now we've come to a more optimal sized animal. And what we can see, and this is a really interesting chart, I think, is the total number of head of cattle in the United States is shown in blue, and it goes to the left axis. So in 1961, you see an increase, and along with that, you see the overall yield of red meat, or the production in million tons of beef, and they go kind of hand in hand. And so we see an increase through to about 1975, and then we see a decrease of both numbers of animals and also meat. But then in about 1987, we see this divergence where cow numbers are going down, but beef production is going up. How can that be? There's only one way that can be. You're getting more beef per cow. In other words, we're starting to select for more productive, optimal animals. And so I think it's interesting that today we have 2 million less head than we did back in 1961, but it's been relatively consistent there, about 90 million total head in the United States, but we're producing almost double the amount of beef with those same number of animals. Similar to the dairy, but not as dramatic as the dairy situation. And so 90 million is the number of head we have, and this is a kind of an interesting global perspective where I'm showing the number of animals in blue, again, going to the left axis, and the production in red going to the right axis. And what you'll see there in this particular year, 2016, US was the major, or the #1 producer of beef at about, 11 or so million tons, but nowhere near the #1 country in terms of number of head. So we're at about 90 million head, and that blue line is 100 million head. And you see there are 2 countries with more bovines than that. Brazil is one, but producing less beef with 220 million head. So that tells me that in Brazil, there's not getting the same yield per animal. And then there's another country that jumps out at you there, and that's India, who of course have a religious prohibition on the consumption of beef. And so it's many different factors influence the overall efficiency of agricultural production. It's not all science, but I think this is a really interesting graph. But there's obviously in developing countries, Sudan and Ethiopia, a huge opportunity to increase the yield in those animals and therefore basically produce more food using the same number of animals. And if we can change that yield gap, that would really help to address what we see coming, which is increased demand in animal protein in the developing world. Are we going to do that with more animals or more productive animals? I think the latter is the better way to go. And I think this graph is really interesting from a global perspective too, because it kind of gives you a feel, again, of the impact of religion, and also which countries eat which types of meat. And so I've got the main terrestrial animals here, cattle, sheep, goats, chickens, and pigs. And we can see different countries. Brazil, again, with the biggest cattle population, is a big producer of chickens and beef. The United States is quite big in cattle, chickens, and pigs. But there's one bar that really jumps out at me in this image, and that is the huge amount of pork production in China. And I really emphasize that because there's some issues going on that could be addressed with gene editing that really influence this. And there's about, I think, 1 billion pigs per year born in China. And at the moment, 20% of them are lost before weaning. 20% of 1 billion is a lot of piglets. So there's some real opportunities there to get better health management and disease resistance to basically stop that loss. So this is a graph of the current meat that's consumed around the world, and people are often surprised to see fish as the #1 animal protein, but that's actually true. That's both aquaculture fish and wild caught fish. But pigs, as you see right now, is #2. Chickens are catching up fast. I think they might take over. But that basically shows, relative to the terrestrial meats, the importance of pigs and chickens. Cattle are below them, and then sheep are quite a bit under that. But the pigs have a similar story to tell as beef and dairy do. And so this is looking at 1980 pig production in the US versus 2015. And so over that 35 years, through conventional selection, we've gone from about 9 pigs per year per sow being marketed to 22 pigs per year per sow being marketed. Pigs are incredibly efficient animals. They're really pretty amazing. And if we have a look at all of the metrics associated with that, we've gone from 1770 pounds per sow per year in 1980 over a 35-year period to 4,200 pounds per pig per year. That's a pretty amazing litter-bearing animal. And if you do the simple math on that, and have a look at how much pork was produced in the US last year, effectively, and look at what would have happened if we hadn't had that genetic improvement over the last 35 years. What if breeders hadn't had access to the tools to improve pigs? You can just do the basic math on this and take the productivity back in 1980 and figure out we would need somewhere about an additional 9 million sows in the US to produce the same amount of pork that we were able to produce with 6 million sows in the current time. And so that opportunity cost, I think, is often lost on people, but it's so important because if we didn't have this progress, we would have a lot less production or a lot more animals with the same production. And when we look at the demand that's projected here, and this is basically the different animal source proteins, and we're in 2019 here with the blue line, you can see milk on the bottom there is the blue, is projected to increase, as is all of the land-based proteins. And again, why is this happening this way? It's basically due to small increases in the developing world. So the developing world is shown here in dashed lines, and this is per capita consumption of milk, shown as purple, and meat, shown as red. And what you see is that they're not anywhere near approaching the consumption levels of the developed world. There's a slight increase in intake per capita, but because there's so many people and so many moving into the middle class, when you look at this as total demand in the developing worlds, you see these very rapid increases in overall total consumption in the developing world. And the question is, how is that going to be addressed? And I would argue the best way to address that is by trying to improve the productivity of the animals in the developing world. This isn't a matter of the developed world eating more animal protein, it's the developing world catching up. And in fact, it's probably most likely, from a meat perspective, mostly the monogastric pigs and chickens. And so anything we can do to improve the efficiency of those animals, and beef for that matter, is going to help with that. So that brings me to gene editing then. Can it be the next inflection point? Can it change the slope of genetic improvement in the same way that genomic selection, artificial insemination, and hybridization have been able to up until this point? And this very beautiful set of images shows work that's been done by plant breeders to use editing to alter the way that the branches form on tomatoes, the number of tomatoes that form on a branch, and the architecture of the plant. And that is really the power of editing, is to manipulate these pathways to improve the overall yield, depending upon what our objectives are. And so just a background to editing, because I know you're not all geneticists here, but basically editing enables you to go in and make a precise double-stranded break in the DNA of the chromosomes that make up an organism. And what it does, they're site-directed nucleases. It enables the nucleases to go to a particular sequence in the genome and make a double-stranded break. And then from there on, the cell takes over trying to repair that double-stranded break. And it can go down the left-hand side of the pathway there, which is called non-homologous end joining. It's a very fancy name for trying to stick it back together. And sometimes there's a small deletion, sometimes there's an insertion, and that can inactivate the protein that's encoded by that region, because the triplet base codes that encode the amino acids get out of frame. And so you basically knock out the gene or inactivate that particular protein. Or you can go down the right-hand side, that's called the homology-directed repair pathway, and there you can provide donor templates shown in blue that has homology to either side of the double-stranded break, and the cell uses that to repair the break and can introduce novel DNA that's shown in blue here, which might mean that you're introducing an allele from one breed of cow, Angus, for example, into Holstein, to give it a particular characteristic that you want from the one breed that isn't present in your breed. So it enables you to move alleles from one breed to another. And that's the types of nucleases are meganucleases, zinc finger nucleases, talons, and then the very, very, very sexy and popular CRISPR. But they're all basically a pair of scissors that can introduce a double-stranded break. So that's the technology we're talking about. Where could it be used in animal breeding? And I show here a slide in different species where different traits have been targeted by research groups throughout the world. This is not in the field at this time. And basically you can see that breeders have not changed what they're trying to do. We're still trying to move towards a breeding objective. The breeding objectives still contain the same types of drivers that have always driven breeding programs. So animals not getting sick, improved product quality, removal of allergens, increased yield, increased resilience, increased disease resistance. These types of traits haven't changed. And so I think that I won't go into the details of each particular one here, but more generally, what a lot of these are doing is basically producing disease-resistant animals by inactivating a gene, knocking out a gene that enables that particular disease to be present in that animal. And also there's a number of traits here that are designed for animal welfare, things like removing horns from dairy cows, things like preventing male pigs from developing boar taint through the maturation of their testes, and also groups that are working to try to produce all one-gender animals for industries where the female is the important One, like chickens and eggs. And so those are the types of applications we could use this for if we're able to. And so here is, for example, gene editing for porcine reproductive and respiratory syndrome virus. It's a real issue in pigs. You can see in orange where this disease is prevalent, Europe and America included. And a group at the University of Missouri inactivated a gene that rendered those pigs resistant to this particular virus. And right now, Genus, a pig breeding company, is trying to take that through the regulatory to enable them to sell those pigs in the United States at the current time. There's a group working at the Roslin Institute to try to address this very horrifying disease, African swine fever, which has moved up from Africa and it's got into Eastern Europe. It's now started to spread. It's a very contagious disease with no cure. It's spread now into Belgium. The wild pigs in Belgium have it. And basically what Roslin scientists try to do is move an allele from the warthog, which is resistant to African swine fever, into the domestic pig. And the hypothesis being that that might protect the pig from this disease. And to my knowledge, they've challenged those pigs, and they won't tell me what the answer was. And so, as all good scientists must do, you have to wait for the publication and the peer-reviewed data to come out. So it's a hypothesis, it might not work, but the reason this is really important is because recently this disease has been found in China. And I mentioned the billion pigs in China and how important pork is to the Chinese diet and the number of the billions of people in that country. And this one to me is really worrying from a food security perspective, because if that gets out in the Chinese pigs, it's not going to be very pretty. We're also doing some disease work in cattle, and so here's some Chinese work that's being done to use editing to address tuberculosis in cattle, which is an issue around the world, but is interestingly a problem in UK as well. You can see the spread from 1986 through to 2010. And that disease seems to be carried by badgers. And so there's an interesting dilemma now in terms of badgers and spreading this disease, and people that like badgers and people that don't like TV, and who gets to decide what happens. So that's an interesting one. But as a geneticist, to me, genetic improvement and genetic resistance to disease is a better, permanent, cumulative solution to disease than treating sick animals with antibiotics and chemicals. And I think it's an easy one to argue from a sustainability perspective, because we lose about 20% of all animal protein to disease globally at the current time. There's also a recent paper that came out from the group at Roslin that are looking at whether they can actually use genome editing to enable this thing called surrogate sire technology. And the idea here is that artificial insemination is not easily used in some industries. Is there a way that we can take elite genetics from a donor male and develop somatogonial stem cells and transplant that into the gonad of a recipient male who has been genome edited to no longer produce germline cells? So an otherwise infertile male is now producing semen that is coming from that superior sire. And you can imagine in this industry, but also the extensive industries like cattle and sheep and goats, that this offers an opportunity to introduce artificial insemination into industries that can't otherwise use it due to the extensive nature of their animals. And to the breeder's equation earlier, what this does is enable higher intensity use of the very best sires. Basically, it's artificial insemination on legs. I'll talk now about some research in my lab. So these are the gene-edited polled calves. And I'm going to let a video that was made by Cornell Alliance for Science do the talking for me, because we developed this as a public outreach piece to explain why we did what we did. If you look at our dairy cows and our beef cows, there's a lot of differences between them. So dairy cows are being bred to be very optimal for dairy production. And as it happens, that brought along the horned phenotype. In other words, dairy cows grow horns. If we look at our beef animals, they've been bred for optimal beef production. And as it happens, Angus, for example, is a breed that doesn't have horns. And we don't really want horns in dairy production systems because the animals hurt each other with the horns and hurt each other with the human workers.
It's a widely accepted husbandry practice that we dehorn cattle, both for the safety of the cattle, as well as for the safety of the people who take care of the cattle and work with them. The way that the disbudding procedure is done here routinely is we will administer a lidocaine block. Lidocaine is a local anesthetic similar to what you might get at the dentist's office for a procedure that might be painful. And then we use an electric dehorning or disbudding instrument that provides heat around the horn bud to kill the cells that would grow the horn. Consumers are concerned about the way we care for our dairy animals, and they expect us to do a good job of providing care and being concerned about animal pain when we have to perform procedures. So I think it's important that we keep those factors in mind and do the best job that we can to try to minimize pain and discomfort.
We're working in collaboration with a company called Combinetics, who had a vision to try to use precision breeding or genome editing to introduce genetics from Angus cattle to the Holstein dairy breed to produce dairy cattle that don't grow horns. And so we haven't altered anything about all of the genetics that makes them great milk producers and all of their Holstein genetics is still in place. What we've basically done is a very precision technique of just introducing one trait that we want, which is polled or horns, and we haven't altered the rest of the genetics.
With the change in technology, it looks like that we can achieve the goal of having calves born without horns and not have to give up the genetic capability of those animals. So it looks like a win-win situation. We've still got very productive, efficient dairy animals, and now have the opportunity or possibility of no longer needing to dehorn or disbud dairy calves.
So I think we have at least one person in the audience that knows that there are some polled, hornless cattle that exist in the dairy cattle population, and you might ask, why not use those? And we just recently modeled what would happen if we used the existing polled genetics to introduce hornless into the US dairy cattle population. And the reason farmers don't use those animals is because they have much lower genetic merit. And what this shows is for Jersey and Holsteins, Jersey's the brown, Holstein's black and white, the polled animals, the big P, are actually, the polled animals are about $150 lower in net merit, which means on average, every calf born to those bulls will produce $150 less profit for the farmer. And so there's a very real disincentive to use those animals, and you can see there's a very small number of them. There's only 3 in Jersey and 19 in the Holstein, versus the much better genetics that are associated with the horned animals. And so that's why farmers tend to use the horned animals, because that's a very big economic hit. And what is also concerning is those animals originally probably came from a beef animal getting introduced into dairy genetics, but they're quite a small pool of animals, and we actually modeled what would happen in terms of introgressing that particular trait from the existing genetics that we have versus being able to genome edit the top 1% of the bulls with good genetics, but just putting in the trait of not growing horns. And what is shown here is the green is the current status quo. So the change in horn frequency is virtually none if we continue on as is, because farmers will keep using the horned genetics. If we say you have to use only those animals that are homozygous to not grow horns, then you'll see a decrease over the 20-year period in the allele frequency, and that's shown by the blue line. But what you see is a really horrible increase in inbreeding because those animals are also closely related to each other. We would very quickly dramatically reduce the genetic variation in our dairy cattle breeding populations if we had to do that. And that's a real problem because, remember, rate of genetic gain is proportional to variation in our population, and so that inbreeding would go right up as shown by that blue line there. If, however, we can edit the top 1% of bulls, we can achieve the decrease in allele frequency. We have a slight increase in inbreeding, that's the red line, but our rate of genetic merit continues on as if it was the control situation. And so really, that's a better outcome than doing nothing or using the existing hornless animals because of that hit you get in breeding. So this particular edit was done using talons as our pair of scissors, not CRISPRs, because it was done a while ago. But what was done is that the homozygous recessive horned allele, you can see here at the pole gene, that 10 base pair little p was replaced by a dominant 212 base pair allele. That is a naturally occurring genetic variation that was done by nature that happened many centuries ago in some of the beef breeds. And so a 10 base pair sequence is replaced by a 212 base pair sequence. Interestingly, not even in a coding region, it's in upstream in a regulatory region. And that makes a dominant trait. So if you inherit that 212 base pairs, you won't grow horns, whether it's heterozygous or homozygous. So that bull, the young bull that you saw a minute ago in the movie, has now grown up, and we've collected semen from him. He is homozygous for that 212 base pair, big P, and you can see there some of his offspring where he was mated to horned animals, so they carried the recessive allele, and all of the animals did not grow horns. Well, Mendel would not be surprised by that result, but we had to actually prove that this is actually what happened. And so we now have these offspring, and they've been quite popular in the media. And so I had an interesting situation recently. We had Wired magazine wanted to do a feature on these offspring, and so this is the female heifer that is the offspring of the edited bull before she came in for her Wired photo shoot. And you can see there she's been made up for her glamour shot. She's had her ear tags cleaned up and she's all ready to go. And then you can see here the photographer is got black screen behind her and is getting every angle possible. And there she is in Wired magazine. So it's quite an interesting transformation. But the one that was even more kind of disarming was that's what she looked like on the cover versus what she actually looks like in real life. So she had a bit of photoshopping there, I think, to make her look a little bit more sinister or something. But she's just a polled cow. And there has been some modelling done by the Roslin Institute showing what could happen if you could combine genome editing with our current selection programs. In other words, genomic selection and our current approach with the ability to introduce useful alleles using editing. And they show that you can, yes, get a step change in the rate of genetic progress if you have alleles you can edit that contribute to quantitative trait variation. So this has the potential to actually be a step change. And I guess to me, I see it as a cherry on top of the current breeding programs we have in place. This will not replace conventional selection. It won't replace records. It won't replace genomic selection. It enables us to introduce useful alleles without linkage drag and to potentially bring in useful novel genetic variation from other species which would be a genetically engineered approach, or from within our own species, which may or may not be, as I'll get to now. So the question is, will we be able to use this technology in animal breeding? And so I'll sum up with kind of where we are regulatory-wise. So in 2018, the USDA, United States Department of Agriculture, which is responsible for regulating genetic engineering in plants, came out with a statement as it relates to gene editing in plants, saying that if you could have achieved that particular genomic alteration using conventional breeding, in other words, through mutagenesis or breeding, crossbreeding wild relatives, then it's not going to be regulated any differently to conventional breeding. It's not going to be in a different bucket, if you will, which gave us some hope that if we were doing a knockout or an allele swap between breeds in a species, that also would be treated like conventional breeding. But in January the 18th, 2017, the FDA came out with a guidance that says all genome-edited animals whose genomes have been intentionally altered are going to be regulated as new animal drugs. And that's the approach that was taken for genetically engineered animals, where you introduce DNA from a different species, but was unexpected for these two bulls to be considered a drug when they have an existing genetic variation that we easily can bring in from the Angus breed. And so they went overnight from being polled bulls to 1,000-pound animal drugs. And I don't think that's a very risk-based decision. I think that's very much a process-triggered decision and is very contrary to how we're treating plants in the United States. And so now, depending upon which kingdom I'm working in, I can use genetic editing or genome editing in plants and not go through additional regulatory requirements if I don't introduce traits that could have been bred into those plants. But if I use that technology in animals, irrespective of the change, it can be as simple as a snip. Remember there's 85 million snips between different bulls? One snip intentionally introduced into cows is going to be regulated as a drug. It doesn't make any scientific sense. But more concerningly, if we start looking around the world, we have regulatory disharmony. So Brazil has ruled that our hornless bulls are not drugs. They're not GMOs, and they're not going to be treated any differently to conventionally bred animals. Similarly, Argentina has come out with their regulatory framework and have asked the question, is there a new combination of genetic material? And if the answer to that is no, and there's no vector backbone in the animal, then it's not going to be subject to regulation as a genetically engineered organism. This is very similar to what the USDA announcement is with plants. In other words, if it's an existing allele in that species, or it was something that could have been produced using conventional breeding techniques, it won't trigger any different regulations because it's just going to be treated like conventional breeding. Then it gets complicated. So Canada has a product-based regulatory system where if you bring a novel attribute to a product, then that's the trigger for regulations. In the case of not growing horns, that's not a novel trait. We've had that in our breeding populations for centuries. So that would not trigger additional regulations in Canada. And in fact, the company Recombinetics that developed that bull has signed an alliance or an agreement with CEMEX, which is a bull co-op in Canada, to introduce this trait into their elite dairy genetics because they want to introduce what I mentioned earlier, elite animals that don't grow horns. So we've got a yes in South America, we've got a yes in Canada. Australia, where I'm from, has a very unusual approach. So if you don't use a template DNA, in other words, you go down the left-hand side of the pathway, the the non-homology end joining, that's not going to trigger regulation because that's effectively mutagenesis. If you use a template, so you come down the homology directed repair pathway, the trigger is going to be invoked if you use long template with no further explanation as to what's long. And so how long is a piece of string? It sounds like a riddle from Alice in Wonderland. So I don't know if 202 base pairs is long. It might be short relative to the 3 billion base pairs that make up the genome, but I imagine it'll be very controversial. Some people will think 1 base pair is long. Others will think much bigger than that. So that's going to be difficult. And then, of course, I assume that everybody in this audience is aware that the European High Court ruled in July 25th, 2018 that edits are going to be regulated as genetically modified organisms. And in their ruling, it basically came down that if it follows that organisms that come from genome editing fall within the scope of the GMO directive, and are subject to the obligations laid down by that directive. But, you know, what for me is complicated in that is that these polled allele does occur naturally. And so the reading of this directive is that organisms obtained by mutagenesis of GMOs, insofar as the techniques and methods of mutagenesis alter the genetic material of an organism in a way that does not occur naturally. But it does occur naturally, so it's difficult. It's also, I think, as you all know, it doesn't apply to traditional mutagenesis done by ionizing radiation, which introduces literally thousands of uncontrolled, unknown mutations throughout the genome. And so, from a scientific perspective, we're in a very uncomfortable situation where, depending where I am in the world, will determine whether or not my polled bulls are drugs, GMOs, not regulated. It's almost inconceivable to me that in 2019, after 30 years of experience with recombinant DNA, we could be this messed up amongst all of these major agricultural producers. And so I think that, to me, spells a little bit of regulatory chaos and trade disruption. And so it's unclear to me what the future of this technology will be, given this level of regulatory disharmony. So in summary, genome editing offers an approach to precisely knock out undesirable traits like disease susceptibility, or precisely introgress desirable traits like not growing horns into food animal breeding programs. It opens up new opportunities for animal breeders to address critical problems such as disease resistance, animal welfare and resilience, and product quality traits. Currently, there's a patchwork of proposed regulatory approaches for the use of gene editing in food animal species, which to me is just a recipe for trade disruption. And I believe that harmonising the regulations associated with this technology in food species in general, be they plants or animals, is imperative to allow both plant and animal breeders access to these tools to basically introduce that next step change that I think is absolutely essential to address improvements in our breeding programs. And the traits that we're targeting are things that I think align with sustainability goals: disease resistance, climate adaptability, food quality attributes. And so we're at a very uncomfortable junction, I think, as scientists, because I don't think what's proposed is very sensible, but what's the course of action that we can take to enable the use of safe innovation in our agricultural production programs. So thank you very much, and I will thank you for inviting me.
So thank you so much, Alison. So now we can take some questions to yes, Jean-Paul? Do you have a microphone?
I can hear. Oh, you need it for the recording, perhaps? There's one right there. Oh.
Dr. Alison, thank you for this stimulating presentation. So I have several questions, but the first question is, so you say you have the offspring from the hornless bull being tested now.
Yes.
Yes. So the bull is Spotify, or is it the other one? Or?
Actually, they were clones, and so it's actually Buri was the one that is the father of the 6 offspring.
Okay. And so when you mean they are tested, does that include testing about non-controlled events that could occur when you edit the gene with the sequence?
Off-target effects?
Yes, off-target.
Yes, actually.
Even more events. This is included in the testing, and what are you doing?
So we have sequenced them all to 20x, whole genome sequence, and the bioinformaticists have run that relative to controls that were basically horned Hereford females bred to horned Hereford males giving birth to horned Hereford babies. And then also we used Buri's dad is a purebred Holstein. We used him to also mate to horned Hereford cows at Davis to have as best contemporary controls as we have, because when you cross a Hereford and a Holstein, you get a lot more genetic diversity because of the millions of SNPs that differ between those two breeds relative to the horned Herefords. And so we've sequenced all of those animals to 20x and have run them through bioinformatic analysis. And the thing is that we can look and see, is it different to controls? But there's a spontaneous level of background variation that is spontaneous de novo mutations, genome sequencing errors, pipeline deviations, that it can't be brought to a decision above there's no more variation in the offspring of the gene-edited animal than there is in the offspring of the controls. It's almost like substantial equivalence for genomes. So that.
Do you plan to go further on with this approach, using the offspring, the descendant from this animal when they are in breeders?
No, we don't. No, we don't. So I don't know if you noticed, but those bulls are not purebred Holsteins; they're crossbred Holsteins, and now they've been crossed to a beef animal. So they're not much use for anything, and so we are going to finish the experiment with the offspring. I guess at this stage, it's a little bit of a fishing expedition. What is it we're looking for? Because I can design an experiment for that. But when it's unknown, it's a little bit hard to design a sensible experiment. So what we did is phenotype the animals, their health, their genomics, and that's as much as we're going to do.
Yes,
with this.
The unknown effects are a matter of concern here in this country.
The unknown effects.
It's quite prudent, so we have to check for a long time before we can really conclude that there are not uncontrolled effects.
I mean, I guess I'm not sure how you would do that. Like knowing, so asking for unknown effects in the future it's a really hard thing for science to do. Like we can't guarantee anything in the future. So what is the appropriate level of that? Do we do this with normal cows and bulls? This type of work, I don't think we've ever really looked at the genomic variation in terms of food safety issue between breeding programs. Is a Belgian blue safe to eat relative to a Holstein? We've never really looked at that type of information from a molecular level. And so I understand what you're asking. I'm also asking what would be acceptable, and I guess I'll put the question to you. What would you propose should be done?
Well, I am not asking that, it's debate at the moment, phenotyping, okay, but genotyping during several generations, and then what looking at, very difficult to understand.
I'm with you, but just to be clear, several generations, that's like 20 years.
Yes.
For each bull, because if we take...
as a starting point before allowing gene editing to be used in a routine manner. Yes, this is more or less the idea.
We do, I must admit, we have the female, she is pregnant, and we were going to, because we've been asked to get milk composition, although I don't know what pole does to milk composition, but we will get milk from her, so she'll have one more generation of offspring. But this is very, very expensive, as you can understand, because every animal, because it's a drug, cannot enter the food supply. So instead of selling them as we normally would, I have to buy them from the university, and then I also need to pay for the incineration of these animals. And so it's a multi-$1,000 experiment. And I have a bit of an ethical issue in terms of what is it that's wrong with these animals that they have to go and get incinerated, and what is it we're looking for? Because there's no biological hypothesis. So I feel there's not an easy answer to these questions. Yeah, and the two bulls, to be honest, they were getting a bit aggressive, and they're no longer with us, and they were incinerated.
Another question about
just.
I'm a veterinarian. I have a question about the horning in the cattle. Once the horned cattle are a lot more difficult to handle because they have no horn. Do you agree with what is the opinion of your veterinarian? Because horns are tools for the vet to handle the cows.
But
if you wish to inject or to make an intravenous injection, you have to secure to handle the cow in certain positions that makes injecting possible.
So typically,
with no horn, it's a lot more difficult.
So it's typically dehorning is done on all of our dairy animals, and to do what you're saying, we put them in head crush, and that's. I've never seen a vet at the front end of a horned animal, to be honest with you. I've seen a lot of vets at the back end of animals, but not at the front end of a horned animal, using the horns in that way. Yeah.
The pollen allele is a dominant allele?
Yes.
Is it fully penetrant?
Yes.
And another question, you mentioned in your talk that it should be possible in the future to make gene editing of the kitty of the quantitative traits. But those genes that are making up the kitty are different kinds. So they are not like the usual genes we refer to with the regulatory region, the coding region, and so forth. They are another kind of gene. Then I'm not sure it's for tomorrow.
So that particular paper from Roslin, they modelled. So QTLs or quantitative traits are made up of many, many genes, but some have a bigger effect than others. And I forget the exact proportion of genetic variation that was explained, but it was some set level, let's say 1%, was what they modelled in that experiment. And that's not an easy thing to find, but there are examples. For example, DGAT in dairy cattle production is one.
Not many.
Yeah, no, I agree. And so
even in the mouse, the kittiel, the anatomy, the constitution of the kittiel is not yet known.
Yeah, I don't disagree, and I think that it hasn't been easy to find those types of QTLs. But I also don't want to say that we won't dissect it down in the future and be able to find maybe 10 that do explain 1% of the genetic variation of traits. I think it's possible that will happen.
The Swiss people had a votation, a referendum about the horning in the cattle. Do you know the results of this votation?
Yeah, it didn't pass.
Good.
Thanks, Alison. I enjoyed the talk, and it was very stimulating to see all your slides and thoughts that you presented to us. Thank you very much. Now, I have obviously a problem. When I discovered that your animals were considered as drugs, X was more than a shark. How can an animal be a drug?
Me too.
It's nonsense, isn't it? You did say that, and I agree. Now, what are you going to do about it? You're not going to limit it. It's just impossible that we can be prevented of all the advantages of those techniques. If animals are considered drugs, it will cost so much that there's no possibility that we stick with that. Not talking about the European decisions, which is another problem. We might discuss that later. But anyway, my question is, what are you going to do now in the U.S. to try to reverse that in the right direction, which would make sense?
So, yeah, so scientists aren't really lobbyists, are they? So we actually have written a petition, a Go petition, asking scientists to sign on that this shouldn't be regulated this way, and that we want harmony with other food kingdoms like plants. And we've written a perspective piece in Nature Science of Food, and those two together we're going to send to the White House and the FDA commissioner. You know, what impact that will have, it's a little bit of an unusual political environment, is a fair thing to say. And so it's a bit unclear, but I think, to be honest, what will happen is the technology will move to countries that have science-based regulatory. And so I think Brazil, South America as a bloc, and well, China's an unknown, but yeah, I think, and so maybe that's where the food security and population growth is. Maybe that's okay.
Well, I say that you just forget about trying to change this direction, and that there's no way that you...
Well, we're doing everything that an academic scientist can, so starting a petition isn't usually how scientists behave, but quite frankly, with these cars, that's going to empty out my bank account, burning all those cars. And I cannot afford to do this research if I have to burn all my animals. It just isn't going to work.
Did you try to approach some members of the Congress and those politicians that might be having your ear anyway? You know, I know that this political situation in the U.S. is very special right now, but I mean, you do have to have people that could understand the logic of it and the base, sound scientific basis to that. Do you have any means to try to get some? It's very difficult in Europe. There's no way that you can convince the politician now, say tomorrow, oh, go ahead, do that, no problem, impossible. Yeah. In the US, do you have that?
There are some, obviously, politicians who have recombinetics in their state. Aqua Advantage salmon facilities in their state are those companies that are talking to those politicians. But as an academic, other than this petition and doing educational workshops on the Hill, that's really what scientists can do in this regard. Bring the technically correct information to enable decision making to occur. And so I don't have an
here, for example, it's very modest, but the Académie d'Agriculture de France has the possibility to make a statement and a position paper saying that it's not sounding based on science. Okay, I'm not sure what the effect of that will be, but do you have that kind of approach in the US right now? I'm not aware of it, but you might have it.
Yeah, unfortunately, there's not that many people working in food animal editing, and so we're maybe 3 or 4 labs in the entire country, and so even though animal ag has this huge environmental footprint, there's just not a huge group of scientists that work in this area. There's a lot that use it in model animals, rats and mice, and it's really hard to get them interested and involved and engaged in these policy issues because it's not stopping them doing their basic research. And so maybe we need to get it to apply to mice, and then we might have a bigger group that's concerned about the impact of this on biomedical research. But at the moment, it really affects the food animal researchers the most.
Thanks, Alison.
This gentleman behind you has been
trying. Of this question, maybe I ask you about African countries. You have shown how important it would be for them. What is their position?
So, all I can tell you is the African coalition has signed on to the like-minded countries as it relates to plant genome editing, where basically, if it could have been achieved using conventional breeding, they haven't really put their toe into animal editing at this stage. I think one kingdom at a time. But obviously this has huge implications for them. Really, animal biotech or animal genetic engineering hasn't been part of their discussion up until now. But it will obviously have a potential impact to them. Yeah. Yes?
Yes, I didn't catch it. If you give us some information on what happens in China or in India about regulation with engineering?
I'm not actually... China is difficult to know what's happening there, to be honest. I can tell you they've trained a lot of scientists because we've all had them in our labs as postdocs, and they are doing a lot of work in this. If you look at the CRISPR publications, the #1 country is China in terms of number of publications. I'm not a rocket scientist, but I can make a pretty big guess that they're going to eventually flick the switch and use this technology in their food production, and that's fair enough. They've got a very big population to feed. India's got the same problem, and so I don't know their regulatory. They've had a lot of pushback on genetic engineering. What the pushback's going to be on editing is unclear, but they're obviously two huge countries, and the decisions they make are going to be very impactful. But I think what's distressing to me as a young country, looking at that slide I showed you, this one, you know, what's Africa meant to do with that? What's India meant to do with that? They're not getting sensible guidance from countries that have experience with this technology, and it's mixed messages. I guess it's almost a 50-50 split. And so, you know, throw a coin. But I think that actually really concerns me because it's not clear what will happen next, and I fear we'll lose the technology.
Any other questions? Well, I have another quite different question. You have a very nice slide. The title is
editing as a chair on top.
Yes.
So, how do public react to this slide? I imagine it will be quite different here in France and in US.
Yeah, I mean, normally, to be honest, that's not a slide I would show to the public because for the most part they're unaware of all the other things underneath the genome editing, right? I assume scientists are more aware of the fact that we do use all of these technologies, but I will say that I have done public outreach events with the horned and the hornless animals, and we've taken the public in. We've done a group of 10 questions before telling them anything. Then we talked about what we did, and then at the end we re-polled them, and we took their survey responses again and looked at what happened. This was done at UC Open House, so it was probably a little bit more of a science-minded crowd. But at the end, when we asked them about genome editing for not growing horns in dairy cows, we had more than 90% that were strongly supportive or supportive. 90. So, and the thing that I think is important is we presented the trade-offs. So this is how we currently do it in terms of the genome, the horn removal, and we do that to protect the animals and the workers. And we could do it genetically, or we can continue to do it this way. Which you kind of have to have that trade-off, or else otherwise they don't even know that we remove the horns now. And so if you don't know that, then you'll say no, because the trade-off isn't obvious. Okay.
Thank you, Alison. I would like to know if you have some tests with readers and farmers regarding this kind of count.
Have I asked the yes, I have asked the animal breeding community because that's who I work with. So the the pig producers are very keen to use this technology because they have the obvious, the PRRS pigs, the surrogate slayers, and also the African swine fever diseases that are coming up. So they have been very vocal and have gone to the Hill and said we want access to this technology, and what you're proposing doesn't make sense. The cattle breeders, the dairy guys, are hesitant because of what happened with recombinant bovine somatotropin. So they will not do anything that they fear will tarnish the mother's milk reputation of milk. The beef guys are less, there are other applications that would be useful for beef production, so all male animals would be more efficient for terminal slayers. And I've had actual breeders ring me asking me if I can do their animals. So I think there's interest there, but not a formal position at this stage. And obviously the artificial insemination breeding co-ops in Canada, for example, are interested in using it because they can, and the Brazilian animal industry sees this as a clear competitive advantage for them, and they will use it because basically they're going to be able to use this technology when I think it's possible that America won't be able to and Europe won't be able to. And then we'll have an interesting situation, because what do we do about imports of semen, milk, meat from countries where an animal has a SNP change that renders them resistant to a disease, but no other traceable DNA? What's going to be the regulatory status of that animal? According to the FDA, there is an allowance for drugs that get used in other countries that are not approved in the US. There's a tolerance. Let's just say it's 1% of whatever. Well, a SNP is 1 of 3 billion. It's going to be 0.000. It's not going to trigger the tolerance. So I think we'll be able to import it if we're going to continue to pretend that DNA variation is a drug.
That is a very important point, as you just mentioned. I believe that I was saying to my friend earlier on, it is not unlikely that, for example, the pig breeders will have to import those size, let's call it this way, from China or South America. Because the comparative advantage of those animals that will be resistant to African swine fever or any other disease will be such as high that if they can find them in Europe, if they can find them in North America, they would go and get them there in China, which would be a complete disaster, for that industry, in thinking that they will have to go and buy those size, those genetics that will, gene all the future generations. It's crazy.
Mm.
But yeah. Logical, right?
Thank you very much for your clear presentation, clear and very clear English for us.
Oh, good.
Very easy to understand. Thank you very much. It was exciting. Well, if you want to meet some people from journalists for the press conference.
Yes.
We are ready. You are ready to put some questions?